In this tutorial, we're going to cover dilutions. Specifically, we're going to talk about dilutions relevant to the experiment you've been performing in the Discovery Lab. A lot of people are unclear about how the dilutions came to be, so we'll walk through some examples, starting with where the compounds came from. Before you started your independent projects, we all tested a compound library that was made available by the National Cancer Institute. What you're looking at on the screen is a plate, a 96 well plate. The compound libraries are given to us on 96 well plates. In this case, in our case, the library that we tested had about 9 plates. Every well has a different stock of a compound that we knew had some activity on, or some effect on cell growth. The compound stocks are 10 millimolar each. When I gave you your compounds, you received a PCR strip tube of 8 compounds. And what I did is I pipetted directly out of this library into those tubes. So the concentration that you got was 10 millimolar. You took those tubes and added 3 microliters of each compound to 3 milliliters of Drosophila media, food. This is a 1 to 1,000 dilution. So the concentration of compound in the food was 10 micromolar. Now these numbers I know are not so easy when you haven't applied general chemistry very much in a lab. But I want to remind you that dilutions are very common. You've been doing dilutions probably your entire life. Taking your stock compound from the library and adding it to food is really no different than making orange juice from a concentrate. You have orange juice concentrate, you add water, it dilutes the concentrate to something that's drinkable. In this case, we take our compounds, we dilute it with food, and we put it in a concentration. It becomes a concentration that the flies can eat. Said another way, if you have a stock solution, before diluting it and you add water, you have the same amount of stuff in there, it's just more diluted. And in chemistry terms what we call water or food is the solvent. To figure out the exact concentrations that you start out with or that you end up with, or to figure out how much volume you need to add to something to get a different concentration, we can use the equation M1V1 equals M2V2, or MIVI equals MFVF. You may have seen this in a number of different forms, but we'll use this one for now. M is the molarity usually, or the concentration. And remember that concentration can be molarity, but if you have a percent solution, percent also indicates a concentration. If you have something that's 10 milligrams per milliliter, milligrams per milliliter is also a concentration. So that M can be replaced by a number of different units. If we go back to our example, what we can do is plug in the numbers that we know. Now, say we start with a compound library that we know is 10 millimolar. We know that because the National Cancer Institute told us that. I'm telling you that what you should do is take 3 microliters of that compound and add it to 3 milliliters of food. And you might then ask, well, what's the concentration that the flies are actually eating? What you can do is plug in the numbers and then solve for the number of millimolar. So in other words, you start with 10 millimolar, you added 3 microliters of that to 3 milliliters of food. The thing that you might notice is that the units in this first example don't agree. So we change the units so that they agree. 3 microliters becomes 0.003 milliliters. And when we solve for that question mark to figure out what is the concentration in this food, it turns out it's 0.01 millimolar or 10 micromolar. Now, this is what you did at the beginning of the semester. Let's apply this to what we've been doing in the lab. So we use colchicine as a positive control. Colchicine is a very old chemotherapy that's extremely toxic. It's derived from the crocus plant. And we can do the exact same thing. We can fill in the numbers. The stock of colchicine that I make in the lab is 50 milligrams per milliliter. I could say to you, I want you to treat the flies with 50 micrograms per milliliter. Your food is 3 milliliters. These are things that we already know. But in order to do this, the thing that you don't know is what is the volume that you should put into the food. So you can solve for the question mark VI, and what you get is 3 microliters. In another example, some of you in your independent projects worked with oils. Now, oils are tricky because oils are presumably 100%, and that's the concentration. So if somebody asks you, what was the concentration of your lavender oil? You would say, well, we started out with 100% stock, and that 100% is MI, or the concentration that you started with. You want to test it between, say, 10% and 2%. A lot of people tested 10% because that's really an extreme amount of oil. But you can imagine that if you were going to take someone's blood and replace some of it with oil, 10% would probably be the upper limit of what that person could tolerate. So people who tested oils would have done something like 10%, 8%, 6%, 4%, and 2%. So how do you do that? We know that we have 3 milliliters of food. I know that I want to start testing my oil at 10%. So I solve for the volume that I need to add to that food to get to that. And it turns out that if you solve for VI, or the amount of oil you need to take out and add to the food, it's 300 microliters. Some people worked with tablets. This was another sort of difficult situation where, in this example, tamoxifen is a known chemotherapy, but let's say we wanted to test tamoxifen. I get you 20 milligram tablets. We know that every tablet contains 20 milligrams of tamoxifen, but we probably want to test that as a solution. It's just easier to pipette than to measure repeatedly. So let's say we take that 20 milligram tablet, we smush it up, and we put it into 1 milliliter of water. The concentration of tamoxifen in the water becomes 20 milligrams per milliliter. We know that we want to test it in 3 milliliters of food, and let's say that we know a therapeutic dose is probably around 10 micrograms per milliliter. So the question is, what do you do with this stock? How much volume of it would you add into the food to make 10 micrograms per milliliter? If you solve for VI in this case, you would get 0.0015 milliliters. And if you convert that to microliters, which is what we actually measure in the lab, you'd get 1.5 microliters. Another example is people who had a chemical that was available from a chemical company. A lot of these chemicals came as a stock solution with a known molarity. So let's say that your chemotherapy or the drug that you're testing came as a stock of 1 millimolar. So this is the concentration that you're starting with. You want to test it at 10 micromolar. You're testing it in 3 milliliters of food. So how do you do that? You ask yourself, what volume do I need to take from here and add to the food to make it 10 micromolar in the food? When you solve for the question mark or for VI, you get 0.03 milliliters or 30 microliters. So these are a number of different examples that we actually did in the lab. And remember that it's really no different than what you tested to begin with, and no different from making orange juice. You're starting with a stock that's too concentrated. You're then diluting it into the food, and the concentration that's in the food is what the larvae are being exposed to. In a fancier way, this is what we're doing. We're taking a concentrated stock and we're making it into a higher volume, which reduces the concentration. So remember that for these types of questions, you'll just use one general chemistry equation, which has on the left side of the equation the concentration that you're starting with and the volume that you need to add, to the right side, which is the concentration that you're aiming for and the final volume. In our case, the final volume is always 3 milliliters of food. And the concentration that you're given would be something that you either made or something that you were given. The concentration that you're aiming for is the concentration of your solution or of your compound that you want the flies to eat. So go through these examples again and again and make sure you understand it, because this equation is probably the most common equation you'll see in a molecular biology lab.